when you're a student, you're still a bit of a baby, even though you're 26 like I am. You're still a baby. You're still learning about the research industry and other things. So the support you get here in terms of, let's say, tailoring your project, making sure it's on track, also career advice. Like, my supervisors are so fantastic in just sitting down and listening to you about what your goal is, and I don't think a lot of students in academia would necessarily get that. The mechanism of clathrin-coated vesicle assembly by AP180 during endocytosis. All that means, so endocytosis is a process that happens in our brain. It's basically the part of the way that brain cells communicate to each other. And AP180 is a protein that lives in the brain, and it is one of the early points of this communication process between the brain cells. So I'm studying how AP180 interacts with other proteins around it to kick this communication process off. And also I'm trying to determine the extent of stimulation in the brain, so the extent of firing or neuronal activity in the brain required to actually trigger AP180 and tell it to start this process. Like epilepsy, for example. You could say in a really sort of simplified way that epilepsy is the brain on fast forward. It's just neurons are firing uncontrollably. So what it is, it's like the brain in fast forward. So if we know what the natural way the brain works is, such as with AP180 in my project, then we can design a drug to inhibit its action so that we can slow an epileptic brain down. This time next year, hopefully I'll be submitted and done and ready to move on.
I can show you what you want to see and take you where you want to be.